even if there is a translocation in a chromosome, that may not affect any other gene. Therefore, they are all silent, there is nothing happening, even though translocation may still happen. And mutations also have got a variety of other different types of frequencies depending upon which population you look at. And therefore, we are also going to study on frequencies as well. So genomic mutations. Obviously, we talked about missegregation, that is non-disjunction and other kinds of events. And these chromosomal mutations are very less percentage, and that is 1 out of 1700. But actual estimates could be different. That is because we are not accounting for those mutations which are frequent, they are getting eliminated, therefore they cannot be accounted for. Most of these mutations occur when gene mutations occur when DNA replication happens. That is, DNA polymerase is not a perfect enzyme. It has got fidelity, like for example, it has got processivity like 3' exonuclease activity. When DNA strand is being synthesized, if an enzyme puts a wrong nucleotide, if the base pair is not appropriate, the 3' exonuclease activity will remove that nucleotide and subsequently is going to put back new one. But nevertheless, things can go wrong. For example, most of these polymerases require magnesium. If magnesium concentrations in your body is not sufficient, then also errors can happen. For example, if you substitute magnesium to manganese, too many errors will happen. Right? Therefore, such errors are very easy. And there is also DNA damage or repair. Even though a wrong base is put in, still there are repair enzymes in the body, DNA damage repair enzymes, which will take care of that kind of a damage very easily. So truly speaking, occurrence of a mutation is not an easy task, because polymerase can take care of most of these by processivity. DNA damage repair enzymes can take care of that. But in spite of all these defense mechanisms that the cell has, still we get some errors. So these errors are truly, really very, very rare, but they still cause mutations. We just talked about the mechanisms, that is genomic chromosomal missegregation, and that is called aneuploidy. We talked about it extensively in the previous chapters. Chromosome mutation is chromosomal rearrangement, they are translocations. But look at the frequencies. Some of them are a little bit rarer, whereas gene mutations are the rarest ones, because it takes difficulties in terms of truly causing mutation against all these repair mechanisms. Okay? That is why point mutations are rarer compared to whole genome mutations or chromosomal mutations, particularly in translocation. Okay? So how does a mutation can happen? For example, if there is an AT pair, and that AT pair is converted to a GC pair, see, that AT pair is converted to a GC pair, you call it a substitution. And if that AT pair is deleted, you call it a deletion. Simple, it's all basic. On the other hand, if there can be an AT pair and another GC pair can be inserted, that is an insertion. So you have different types. Depending upon the type, let us say that there is a substitution here, and this particular gene is being transcribed, and you have got an mRNA, but that GC is not going to change an amino acid. You call it a silent mutation, it's nothing, no problem at all. Whereas if there is a deletion, let us say, in a coding sequence, that particular deletion, because reading frame could be altered when the mRNA is being translated. But again, let us say reading frame is still altered, and your protein adds another extra 10 amino acids extra, but those 10 amino acids do not have any kind of effect on the protein function, then the deletion is meaningless. So sometimes the deletion can cause a long reading frame, the protein is totally altered, and that could cause a deleterious effect. So it all depends on the context and how exactly this deletion is really functioning at the protein. And insertion, very similar fashion. It can also cause a reading frame shift, and similarly, some of them can cause deleterious mutations and some of them don't. So, nucleotide substitutions, deletions, and insertions, as well as there are what are called dynamic mutations. The dynamic mutations we talked about are those, what are those? Which are constantly expanding, like trinucleotide repeats and dinucleotide repeats, etc. There are mutations called missense mutations, chain termination mutations, RNA processing mutations, and also there are hotspots where these mutations can happen much more prevalently by CG, CPG residues. For example, a missense mutation. What is that? Sense codon, it is there, and some other codon is, some other amino acid replaced, that's a missense. A chain termination mutation. Suppose, what are the chain terminators? UAG, UGA, and UAA. These three stop codons, there are various reasons why those stop codons can go bad. Like, if there is a small change in those stop codons, that is no longer a stop codon, it can code for amino acid. Therefore, there won't be a chain termination and the protein will be translated even longer. But that doesn't mean, again, phenotypically the protein is deleterious. It all depends on the nature of the protein. RNA processing mutations. For example, if there is an mRNA which is long, that is the heterogeneous nuclear, precursor mRNA, which contains introns, and if there is a mutation in the splice junction, you are going to not process the messenger RNA completely, so you will not have a full complement of the messenger RNA intact, and thus you will get those abnormal proteins based on abnormal splicing. That is because you can skip an exon, and therefore the shorter protein can be generated. Alternatively, you can have a cryptic splice site, like in other words, a mutation in an intron can cause a new splice site, and therefore you can create an additional exon, and thus the protein can be increased in length as well. Thus, all these three mutations will cause definitely some deleterious nature of mutations, as well as sometimes it doesn't have to cause. Right? So you have to look at it very carefully. So if you have methyl cytosine, that is a CG residue, and therefore you can demethylate it, and that will become, demethylation and remethylation can convert C to T, and therefore because of that, when the C is converted to T, that T can, when it is being copied, that can put an A, and therefore the transition can happen very easily. Okay, so that is one of the ways that CPG residues will cause mutations in those regions. And in addition to point mutations, you can also have extremely small deletions, like a small codon, a small triplet or quadruplet or whatever. So, and also large deletions can happen and insertions, okay? And this is because of a recombination phenomenon. For example, I think we talked about it, but now we are going to go to methodically the mechanism. If you have two copies of a gene, or two segments of DNA which are exactly copies like that, and if one chromosome crosses over with the another one, due to unequal crossover, you can have a segregation and give you a generation of three copies and deletion of in another one deletion. So the same situation, that is, it is between two chromosomes, whereas even within the chromosome, for example, if there is only a single chromosome, it can fold back because these repeats are very similar. So it can fold back and give you a crossover, and therefore it can generate an altered form of the original structure. So these are deletions and insertions because of unequal crossover or unequal recombination. We talked about these mutations in Fragile X and Huntington. These are called dynamic mutations because they are constantly changing from one generation to another. Okay. This table is with you, so I don't have to, it's very difficult to copy this table. So, if you look at the percentages of these guys, this is the missense mutations are the ones which are really highly prevalent. RNA processing mutations are only a little bit less than those. Nonsense mutations are also going to be rare because, most of the times it becomes lethal and therefore nonsense mutations are rare. Splice sites and also regulatory mutations. What are regulatory mutations? The regulatory mutations are those which are present upstream from the gene or downstream from the gene or present in the UTRs. These are all called regulatory because they are going to maintain the levels of the protein. For example, if the promoter mutations happen or 5' UTR mutations can happen, which we talked about in the last class, like prothrombin problem in thrombosis, either more amount of RNA can be generated from the transcriptional activation or less amount, depending upon the situation, you can either get more or less. But these mutations are going to be extremely rare. So most prevalent are the missense, and then the nonsense and other things are a little bit lesser, but regulatory mutations are even going to be extremely low. So you can think about, because the nature of mutations, you can actually calculate in a population, that's at least a guesswork. Okay, this particular thing is going to be less frequent in a given population. And that knowledge is important because depending upon what mutation, you can actually give a genetic counseling to a patient. Yeah, look, this frequency is going to be low, this is going to be pretty high, so the chances of yourself is going to get a mutation on the next generation is pretty high. That is why this information is important. Everybody knows about this information, but in the context of human genetics, that's what we are going to learn. And then there is a nomenclature here. Okay. For example, C1444, G greater than A. That's the symbol that is. So you can actually tell that that particular base is changed, and there is a change from G to A. Okay. In a similar way, this is IV, IVS, that is intervening sequence 33, that is the number at which exactly it happens. There is a +2G, obviously there is an insertion, and so on. So this nomenclature is also very critical. You need to look into the nomenclature carefully. Suppose if somebody gives you C1524, 1527, that means there is a triplet or quadruplet deletion of CGTA. Okay. So this nomenclature tells that that is a deletion in that position of the genomic sequence. Yes. Because C is also being deleted. That's what it is. So 1277 to 1278, if you put INS, there is an insertion and that sequence is inserted. So this nomenclature you have to pay attention. Okay. And similar way, at the level of the protein, you can say GLU6-to-val, that means the glutamic acid of the number 6th residue in a protein is changed to valine. And same thing here, glutamin 39. Now we can estimate germline mutations. Obviously, germline mutations will vary depending upon the size of the gene. The smaller the size of the gene, let us say randomly a sun ray or ultraviolet radiation is happening in your skin. So if I focus on one particular gene like a globin, the chances of hitting that particular ray hitting that gene is going to be extremely limited because it's a very small size. Same thing, if you put a huge size, like immunoglobulin gene or something like that, which are usually huge, you're going to get mutations in that area because much more random hits are going to be possible. So that's obvious. And there are other factors which are also going to increase germline mutations, that is the age and gender of the parent. I mean, if a person gets married at the age of 85, obviously his sperm mutations are going to be tremendous. And then, the baby will have lots of deleterious features. So that is age and gender of the parent. Like, a woman after 35, you've got issues with So again, it depends on all these things, obviously mutational mechanisms. You know, if you're talking about chromosomal mutations, that is, germline mutations will be different. And hotspots, as I told you, CPG residues are very heavy hotspots. All these things contribute to the germline mutations. So if you take a mutational rate and different loci, see, each locus has got a different mutational rate. It's not going to be constant for all, because why is it that there are some disorders which are more prevalent? That is because obviously some regions of the genome are more prone to mutations than other regions of the genome. Some regions may be having clusters of CPG residues, some regions may not. So if it were to be a uniform distribution of AGCT in the genome, then you would expect the mutational rates to be identical irrespective of the gene. But that is not the case. So there is, although it is a random sequence, but there is still non-randomness in the entire genome. And because of that, if you take a look at Duchenne muscular dystrophy and hemophilia A, and for example, there is not much difference. But this locus, for example, this hemophilia A and B are still present